Good day, this is Professor Will, CBMD PhD. Today's Sunday, August 25th, 2019. It is 2:22 and 57 seconds Eastern Standard Time, and this is a continuation of my scientific research on pathophysiology, going a bit deeper into DNA replication. So let's get started here. DNA strand synthesis. The genetics of DNA strand synthesis are reflected in the DNA polymerase. Acidicia coli is known to contain at least 3 distinct and separate DNA polymerase, all possessing the following enzymatic activities: synthesis exclusively in the 5' 3' direction, utilization of 5' deoxynucleoside triphosphates for substrates, the copying of single-stranded DNA, template incorporation of base analogs of ribonucleic triphosphates at low efficiency under altered conditions, such as in the presence of manganese, a 3' editorial exonuclease that preferentially removes mismatched 3' termini, and a rate of synthesis that does not approach that of DNA replication in the cell. Despite these similarities, distinct physical differences exist, and the cell uses exclusively DNA polymerase III for DNA replication. The synthesis subunit for DNA polymerase III, the alpha subunit, is encoded by the POLC DNA EG. DNA polymerase I, encoded by the POLAG, and appears to be an auxiliary protein for DNA replication. Cells lacking this enzyme demonstrate viability, although those lacking the notable 5' 3' exonuclease activity of this enzyme are only partially viable unless grown in high salt. DNA polymerase I is very important for survival of the cell following many types of DNA damage, and in its absence the cell has persistent single-stranded breaks that promote DNA recombination. DNA polymerase I appears to be a particularly potent effector with DNA ligases in sealing single-stranded nicks, perhaps because of its ability to catalyze nick translation, in which the 5' exonucleolytic removal of bases is coupled to synthesis activity. Neither of the other DNA polymerase appears to possess this property. DNA polymerase II is an enzyme without a defined role in the cell. It has been cloned and overproduced and has been found to bear a closer relationship to T4 DNA polymerase and human polymerase alpha than to either of the other two E. coli DNA polymerases. Nevertheless, it is capable of interacting with the subunits of DNA polymerase III complex. Cells that completely lack the structural gene for DNA polymerase II, the POLB gene, show normal viability and normal repair after DNA damage in many circumstances. Among suggested roles is synthesis to bypass DNA damage. DNA polymerase III is a required replicase of E. coli. The fact that it plays a significant role in DNA replication is demonstrated because DNA ET's mutants contain a temperature-sensitive DNA polymerase III. Despite having properties similar to those of DNA polymerase I and II, DNA polymerase III is specifically required for DNA replication. This is a reflection of its ability to interact with a set of subunits that confer particular properties on the complex. In the complex term, the holoenzyme, DNA polymerase III, takes on the properties of high rate of synthesis and great processivity. Intuitively, processivity may be thought of as the ability of an enzyme catalyzing the synthesis of DNA to remain tracking on one template for a long period of time before dissociating and initiating synthesis on another template. Highly processive enzymes are capable of synthesizing 1,000 nucleotides at a single stretch before releasing the template. DNA polymerase III appears to be uniquely processive among the E. coli DNA polymerases. Holoenzyme DNA polymerase III. In addition to the asynthesis subunit, there are at least 9 constituents of DNA polymerase III, holoenzyme complex. Most of them have been shown to be the products of required genes, as demonstrated by the fact that mutations in the gene produce conditional cessation of DNA replication. DNA E, DNA Q, DNA N, and DNA X of the knockout mutants are inviable. Holo A encoding the sigma subunit and holo B encoding the D subunit. A knockout mutation of holo E did not impair cell viability, implying that the null subunit is dispensable for normal growth. The gamma protein and the T protein appear to be the products of the same DNA X gene. This is a case in which frame-shifting termination of protein synthesis plays a role in producing different proteins from the same gene. Mutants constructed with the frame shift in the DNA X gene that abolish production of gamma, but do not affect tau, are viable. However, tau has been shown to be essential. The beta subunit is a product of the DNA N gene. This subunit appears to confer specificity for primary utilization upon the complex and to increase the processivity. The epsilon protein of the holoenzyme complex is known to provide a powerful 3' editorial exonuclease activity. This is manifest by the fact that, in addition to lethal mutants in this gene, mut-D mutants that show increased error rates in DNA replication, mutators can be isolated. Physical studies as well as genetic studies indicate that DNA polymerase III holoenzyme complex exists in a dimer form. The stoichiometry of various subunits suggests that the dimer is not exactly symmetrical, but it does appear to be symmetrical for the alpha, beta, and epsilon subunits. The holoenzyme comprises two dimerized beta subunits beta 4 and dimeric core pol 3, alpha 2, epsilon 2, null 2, tau 2, in a single gamma complex, gamma 2, D1, D1, X1, sigma 1, that appears to be involved in loading the beta processivity. Clamp onto the DNA template. The physical and genetic evidence supporting dimerization of DNA, polymerase III, is in accordance with the structural model of replication. This is a so-called inchworm or trombone model of DNA replication. As indicated in the model, a dimer at a growing fork would allow coupling of rates of synthesis on the leading and lagging strands. The strand made continuously and the strand made discontinuously, because the strand made discontinuously may require frequent initiation. One might expect synthesis of this nascent DNA strand to be slower. To prevent a discrepancy in growth rate between the strands, dimerization of the synthesis unit for the two strands is a method for locking the rates in step. It is possible to do this by assuming that the DNA template for the lagging strand loops out in such a way as to provide permissible polarity for the nascent strand. A key player in organizing the replisome, that is, the multi-protein complex that replicates the chromosome, appears to be the tau subunit, which has been shown to interact with both DNA B helicase and primate. The coupling of polymerase III to DNA B explains the high level of processivity on the leading strand while allowing the other half of the polymerase III holoenzyme to cycle on and off the lagging strand during micro-initiation. Termination of replication. Termination of DNA replication is complicated by topological problems created by the circular nature of the bacteria chromosome. The double helical structure of the template DNA, given a semi-conservative mode of DNA replication, results in two interwound chromosomes which must be unlinked by topoisomerase. Decatenation of the newly replicated chromosome is accomplished primarily by topoisomerase 4, a double-stranded DNA topoisomerase that closely resembles DNA gyrus topoisomerase 2. Another problem is that bidirectional replication on a circular template must be coordinated so that the opposing replication forks meet and terminate DNA. Synthesis at a defined location, otherwise part of the genome might be over-replicated. A specific region of the chromosome approximately directly opposite the origin on the E. coli map at the 32 min region represents the termination region. This region is particularly sparse in genetic markers. A specific terminator protein, TER or TUS, binds to a sequence called TER or tau. TER sequences are arrayed in the termination region in an inverted repeat configuration such that binding of TUS protein to TER sequences confers a polarity to blockage of the replication fork. TUS protein has been described as a polar contra helicase because its ability to inhibit DNA B. The replication fork-specific helicase depends on the polarity of the TER sequence with respect to the origin of replication. TER sequence with respect to the origin of replication. The replication fork is unimpeded by TUS bound to a TER sequence in the forward orientation, but it is blocked by TUS bound to a TER sequence in the reverse orientation. Because of the inverted configuration of TER sites in the termination region, a replication fork that progresses through a forward-orientated TER site will be stopped at the next reverse-orientated site. The termination region thus traps replication forks, ensuring that part of the genome is not over-replicated. Bacillus subtilis has involved a similar mechanism with a termination protein, RTP, that binds to a specific inversely orientated repeats, also called TER sequences. Of note is the fact that the E. coli TER sites are widely separated by approximately 350 kb. Whereas the innermost oppositely oriented TER sites in B. subtilis are only 59 BP apart. The basic strategy for termination in the two organisms is quite similar, but the molecular mechanisms appear to be different. DNA replication termination may also be a control for cell division. Good day, this is Professor Will, CBMD PhD. Today, Sunday, August 25, 2019, is 3:35:51 seconds Eastern Standard Time, and this concludes my research on DNA replication on pathophysiology. Hope you enjoyed listening. Have a great day.